So yeah, it is recording now.
Okay. Oh, we should share the slide. Yeah. Um,
I think that might actually work. Hmm.
um, and then start.
Maybe you just have to make sure that we're doing that.
It's called.
But you'll be presenting on your slide.
If that's convenient, yeah. If not, I also have a USB.
Okay. Yeah. So we should probably do it on there.
On there?
Yeah,
Okay.
just because we have the Zoom set up.
Sure.
Oh, I should
don't
get to
think
share
the-
her.
Here as well, right?
Oh, you're already in Zoom. Then great.
Okay. Okay.
Yeah. We'll just switch over there.
Okay, I'll get us started. Hi, my name is Yerin Kim. I'm today's BAMBUG host, and we're gonna start February BAMBUG today. Please silence your phones if you haven't. This event is being recorded so that we can upload on YouTube for those who missed the talk today. And starting from 11 a.m. after the introduction, we're going to have introductory training talk and then featured talk. And after the talk, we're going to have pizza and social networking. We are lucky to have the BAMBUG organizing committee. We have several faculty members, Dr. William Seo, Dr. Amy Lee, Dr. Ross Hawk, and we have also volunteers at UBC and also at SFU. We have upcoming BAMBUG events in March and April. If you subscribe to our mailing list, you can get the information about the future talks. So you can scan the code right away if you want to subscribe to with your phone. And we're always looking for a future speakers for BAMBUG. If you visit our website, you can go to the contact and suggest a speaker you prefer. BAMBUG is sponsored by Graduate Studies in Bioinformatics at University of British Columbia and Simon Fraser University, Genome British Columbia, SFU Big Data Hub, Michael Smith Laboratories at University of British Columbia, Langara College, Stem Cell Technologies, and BAMBUG is affiliated with International Society for Computational Biology, Montreal Bug, Toronto Bug. For Zoom audience, if you have any chats, if you have any questions, please write on the chat, and our volunteer will voice it for you. So we have introductory student talk by Delina. She's a PhD student at the Talhook Lab at UBC. And the title of our talk today is Assessing the Reproducibility Crisis of Vagina Microbiome Studies for Clinical Applications in Endometrial Cancer. And after the training talk, we're going to have featured talk, Dr. Ana Conesa. She's an international speaker. She's courtesy professor at University of Florida, research professor at Spanish National Research Council, and also co-founder of Biobam Bioinformatics. And the title of our talk today is Third Generation Long-Read Transcriptome Sequencing for Unraveling Transcriptome Complexity. Delina, please take it away from me when you're ready. Sure, I'll enable the screen with you. You can share the screen.
Hi, everyone. Thank you so much for being here today. I will be presenting our work on assessing the reproducibility crisis in vaginal microbiome studies in the context of endometrial cancer. So for a little bit of background, for the longest time, the uterus was considered to be a sterile environment. But recently, studies have not only confirmed the presence of a microbiome, which includes bacteria, fungi, and yeast in the uterus, but also associated it with various gynecological diseases, such as endometriosis, endometrial hyperplasia, and endometrial cancer. Endometrial cancer is a common gynecological malignancy here in North America, but unfortunately, there are no general screening techniques available, and we're relying on an endometrial biopsy for a diagnosis. So several studies have aimed to identify a vaginal microbiome signature for EC, but none of them have translated to the clinic yet. And this is because all microbiome studies in general are facing a reproducibility crisis caused by a variety of reasons as shown on the graph on the slide. But the ones that I want to highlight the most is that the healthy microbiome is very dependent on individualistic factors such as age, BMI, ethnicity, and vaginal pH. And some of the technical factors that I want to highlight is that there is often incomplete methodology quoted by authors in these papers. There's unavailability of metadata for us to reproduce the results, and there's lack of method standardization. So different analytical pipelines often lead to really different results. So the objective with this study was to evaluate EC vaginal microbiome studies for reproducibility and replicability. We defined reproducibility as using the same data and the same analytical pipeline as the authors, and replicability as using an independently curated data set with the same analytical pipeline. We then wanted to test latest bioinformatics pipelines for improvement to see if we can improve replicability. And we also wanted to test implications of these analytical pipelines on downstream models. So before I go into the methods of the study, I just wanted to give you a brief overview of what a 16S rRNA bioinformatics pipeline looks like. We have sequence reads that will undergo quality filtering to result in some sort of a sequence space. And then you can take the traditional route of clustering similar sequences together, or you can denoise using machine learning and statistical models to remove sequencing artifacts and only preserve the true biological variants.
Is it okay right now?
So for the study design, we identified 5 studies that reported an EC vaginal microbiome signature. And then we implemented each study's bioinformatics pipeline on all the data sets. So the data sets we identified are on the table here. And the pipeline they used is also one of the columns. And then we mentioned whether they've done clustering, denoising, and the taxonomical resolution that they achieved using the pipeline. So almost all the pipelines that have been published so far only achieved genus level taxonomical information. And then we went ahead and reprocessed all the data sets using the data2 pipeline. And we assigned taxonomical information down to the species level, which I'll show how helped us improve replicability. We also use metrics such as alpha-beta diversity, differentially expressed taxa, and interstudy replicability to measure reproducibility and replicability. So the data2 pipeline overview, we trimmed all reads at the 3' end to preserve a minimum quality of a Phred score 20. All data sets were then denoised using the data2 pipeline, resulting in amplicon sequence variants. And then we assigned taxonomical information using 3 databases: the Green Genes database with a uniform taxonomical distribution, the genome taxonomical database, again with a uniform taxonomical distribution. And then lastly, we use the genome taxonomical database again, but this time we gave it expected species weight in the vaginal microbiome environment. And this is what really helped us get the species level information that we were looking for. We then merged the taxonomies using RESTscript implemented in QIIME2. So alpha diversity is a measure that measures diversity within a local community. So either within the benign state or the malignant state. And from the literature review that we did, we know that benign state usually has a lower microbiome diversity in the vaginal environment. It is mostly dominated by a single species, Lactobacillus. And in a malignant state, the rarer taxa become more prevalent, which increases the microbial diversity. Beta diversity is a measure of dissimilarity between 2 communities, so between EC and benign, and it is based on ecological distance. And then we use an ANOVA-based permutation to test for how much of the percentage of how much of the variance in the data set is explained by clinical covariates such as histology, vaginal pH, age, BMI, and ethnicity. So for the results for alpha diversity, The plot here has the alpha diversity metric Shannon index that we used on the y-axis. Each vertical facet is one of the pipelines that we implemented on every cohort on the x-axis. So we have 5 pipelines implemented on each of the 5 cohorts. And the trend that we're looking for is that the red box plot is higher than the green, which means EC has a higher microbial diversity than benign. And what we noticed is that all pipelines are reproducible, but not all are replicable. So if you take the Antonio Walsh pipeline on their own data set, you'll see the right trend. But when you take the same pipeline and process another data set with it, we'll see the reverse trend. So for example, in Temenzi, we see the reverse trend. And then the data to pipeline with species level information is all the way at the end here. And you'll see that the species level information actually helps us achieve the right trend across all the data sets. For para-diversity, we have a similar plot where each vertical facet is one of the 5 pipelines that we implemented on each of the 5 cohorts on the x-axis. And the y-axis has all the clinical variates that we could get access to. And each colored box is the percentage of variance explained by that clinical covariate when processed by that pipeline on that particular cohort. And the brighter the box is, the more percentage of variance is explained. So histology across the board, across all pipelines and data sets explains less than 10% of variance in these data sets. And individualistic factors such as age, vaginal pH, and BMI have a stronger influence on these communities. So BMI throughout in one of the cohorts is almost explained 80% of the variance in these data sets. One thing I want to point out in this slide is that not all studies report all of these clinical covariates. So for example, BMI is only reported by 3 of the 5 studies that we found, which is a problem because these analyses are then confounded by not taking into account BMI. For differentially expressed taxa, we used a tool called ANCOM-BC, which is for microbiome data. We were unable to reproduce results from any of the 5 studies, but using species level information, we found various species of Peptinus phyllis differentially expressed in endometrial cancer in all 5 studies. So the network plot here, histology, BMI, vaginal pH, and age are one of the nodes. And the thickness of the line represents the number of studies that particular taxa was differentiated in. So Peptinus phyllis has the thickest line. It's differentially expressed in all 5 studies. And then we found different species of Streptococcus, Peptostreptococcus, and Prevotella in 4 of the 5 studies. We also wanted to measure interlab lab replicability. And what this means is that we try to learn distinctions between EC and benign on data from one study and evaluate if the same distinctions hold on data sets from other labs. To do this, we use boosted trees and area under the curve as a performance metric. And the plot here basically shows each vertical facet is one of the data sets that we trained on using the pipeline shown on the x-axis. And the pipeline that we are recommending or highlighting, the data to pipeline with species level information, is the red box plot here. And then we tested on all the other 4 cohorts. And we see that the data to pipeline with species level information usually improves performance across the board, or it's pretty similar to the one where it's not performing the best. So in this case, it's not the best, but it's pretty close. In every other case, it's outperforming the other pipelines. So some key takeaways is that species level information improves replicability, and it is needed in vaginal microbiome studies in order to have a robust signature. EC has a higher microbial diversity compared to benign state. We identified different species of Peptinophilus as a potential biomarker for endometrial cancer. And lastly, histology alone only explains less than 10% of variance in these data sets, and studies must account for individualistic factors such as age, BMI, and vaginal pH. We next aim to identify a robust signature or come up with a clinical assay, such as a classifier, that will benefit screening programs. And to do this, we'll do a meta-analysis from all the studies that we found. And we're hoping to collect cell-free DNA data as well that we can incorporate with the microbiome to be more confident in our predictions. These are my references, and I just want to say a big thank you to all members of the Uterine Health Research Lab, especially my PI, Dr. Aline Talhook, and my supervisory committee, and to all of you for listening. Thank you.
Do we have any questions? Okay. Okay, thank you so much, Tulina. Can you stop the sharing? Oh, Kara.
Sorry, I had a question. So for the pipelines where the results were like reversed in the replicability experiments, did you find that like the input data that they built their pipeline on, was it using a different sequencing method to the one that it was testing and reversing on, or was it similar methods?
So they were all similar. Sorry, they were all similar methods of sequencing, but the way they normalized the data was really different. And we think that's what's causing the reverse trends in replicability. So normalization and taking out sequencing artifacts seem to be a major role in why these results are really different. But sequencing was all done using maSigPro, and they all mostly sequenced the region V4 of the 16S rRNA gene.
Okay, thanks.
Any questions? Okay. Delina, can you please stop the sharing? Okay. Okay, so the next speaker is Dr. Anna Conesa. Dr. Conesa, can you share the screen with us?
Yes. Yes.
So we might have to connect the mic here.
Are you seeing it right?
So we like, are you tech?
Do you see the full screen?
No, we're seeing some sort of presentation mode.
Okay, so let me.
Dr. Conesa,
Yeah.
can you? Oh yeah, that.
That's better.
Yeah, that's perfect.
Okay.
Just a second. We're trying to connect the mic here.
We should have quality as well. I can do it.
Hello?
We have nothing.
Can I start, or are we waiting for
Could you
something?
ask her to turn her mic on and speak?
Dr. Conesa, can you
Yes.
can you hear us?
Yeah, I can hear you. Yeah.
So we can't hear her.
You cannot hear me?
Oh, we cannot hear Dr. Conesa.
Oh.
I think it might be an issue with her mic, maybe.
I don't think so.
Oh,
We can hear her on the Zoom.
there.
Hello?
Yeah. Yeah, and we can hear her on the Zoom. So I think it's a UBC connection to the whatever you're presenting. The audio isn't going through.
Yeah.
You speak, please.
Hello, can you hear me?
We can, yes.
So now you can hear me, right?
Yes.
Okay, great. All right, so
We are good at UBC.
okay, so can I start? All right. Okay. Thank you very much for your invitation to give this seminar here today. I'm at the other side of the planet, but Zoom is great for this kind of seminars. So I will be talking about the utilization of long reads transcriptome sequencing for understanding the complexity of the transcriptomes. And you have. Okay, let me see if I can read my presentation. Right. So you may know that long-read sequencing methods like PacBio and Nanopore were declared like the method of the year by Nature Methods in 2022 because of, they have been creating some kind of a revolution both in genomics and transcriptomics research. And when you apply long-read sequencing methods to the analysis of the transcriptome, basically the idea is that it can help you to resolve the contiguity of alternative express isoforms. So you know that genes may express multi-exonic genes, may express alternative isoforms because of alternative splicing. And these are difficult to capture by short-read technologies because they cannot resolve the contiguity of the transcript models. But when you have long reads, you can have a single read that covers all the junctions structures. So you don't need to assemble the transcriptome, but you just can detect directly the alternative isoforms. So this is important because there has been a lot of research, but also controversy on the functional relevance of alternative splicing and alternative isoforms. And there are many examples indicating that alternative isoforms are involved in distinct biological processes or functions. For example, they may control the inclusion of substrates, so that's the expression of one isoform of another will change the presence of an active site. The same with binding to DNA as a transcription factor. This could be controlled by alternative splicing of the presence of protein-protein binding motifs, but also there is the alternative utilization of isoforms may impact the localization, the cellular localization of the protein, which may lead to a different function. And not only at the coding sequence, but also at the UTRs, you may have alternative isoforms, and this may control regulatory elements that affect the stability of the transcripts. Also, the utilization of these long-read technologies have resulted on the annotation of the discovering of hundreds of thousands of novel transcripts, even in well-annotated species, like in human. This paper with Nanopore, they were reporting 70,000 novel transcripts. And in species such as Arabidopsis, long-read methods resulted in more than 100,000 transcripts, and every paper that you find using long reads for genome annotation is reporting really a huge amount of novel transcripts. But actually, if you look at how the data looks like, it seems that it's not as easy to identify these alternative isoforms. And this is a representation of the long reads data captured with different technologies of this GAPDH gene in one human cell line, WCT11. And its line represents a transcript model that was obtained by long-read sequencing methods. And here at the bottom, you can see the gene code annotation of this gene, in which you can find 6 alternative isoforms, in which you can see some exons that are constitutive and some exons that are alternative, depending on the isoform. And this is the diversity of isoforms of transcript models that you find with the long reads. And you can see some things like, for example, here, this is an intron retention event. You can see some exons that seems to be very well included all the time, but also some areas here that seems to be like very noisy or very variable. So the question is whether all these transcript models and all these signals is real, or there may be some issues with the technology itself. So there is a number in the analysis of long reads RNA-seq data. First is the accurate call of transcript models from the long reads. Today, there are already many tools for transcript reconstruction. And as I mentioned before, many novel transcripts are discovered in each new experiment. But also it's important to use this information to investigate the function and the regulatory role of the diversity of isoforms that we can discover by long reads. So how you assign them function and how you look for differences in the usage of transcripts from this data. Can you use the same methods as for regular Illumina short RNA-seq or not? So my lab has been working on these questions for quite some time, and we have been developing tools for the analysis of this data. And today we have this framework we call the Functional Isotranscriptomics Framework that is composed out of 3 main tools that I will explain today. That are related to the quality control of the long reads data, the functional annotation, and the statistical analysis. So the first tool I will be discussing is SQANTI 3, and actually this tool was developed trying to answer the question of what is the quality of the transcript models with long reads sequencing. And this has been a general question formulated by others. And actually, I participated in the longer gas benchmarking projects that was trying to answer this question. So how accurate are these methodologies for identifying transcript models? And in this project, we generated long-read sequencing data for 3 species, human, mouse, and manatee. We have different cell types or combinations of cells, and also spiking data of synthetic RNAs in our samples, or computationally generated data. We use different library protocols because this is also characteristic of the methods. There are now different library preps available. The regular is the cDNA, the full-length cDNA library preparation method, but you can have direct RNA sequencing with nanopore. There is a technology called R2C2 that creates circular molecules that are sequenced by nanopore several times to increase accuracy. And some methods like CapTrap are capturing the transcript from the 5' end, from the 5' cap. And then we look at different platforms, PacBio, Nanopore, and Illumina for comparison. So this is the consortium that put this project together, and I have to thank all of them, especially Gloria was involved in experimental validation. Some other people were part of the gene code and the UCSC genome browser. This person here from a part of is a member of my lab who did quite a lot of the analysis in this project, and most of the data that I am presenting was generated by him. And then we have a number of labs who participated in the challenge. So basically what we did was, before that, so we have different challenges, but the first challenge was to identify isoform detection with a high quality genome. And here we use the human and mouse data, also mixtures. We use also the SIRFs, the spikings, and synthetic data. So here you have the data as it looks like, just the reads, and you can see that already the reads have different features. So here you are seeing the read length, and you can see the 6 different combinations that we have of library preparation methods and sequencing technology. So we have the combination of cDNA with PacBio, cDNA CapTrack with PacBio, and then with nanopore, we have this for library preparation, CapTrack, R2C2, cDNA, and direct RNA. So you will be seeing these 6 combinations throughout this presentation. So what you can see here that there is quite some differences in the length of the reads, with PacBio cDNA giving the largest range and also the longest reads, followed by the combination of R2C2 with nanopore. And other library preparation methods had notably shorter reads. So you can already see that there are some important differences in the starting material. So other differences from this data was the number of reads. So here you have the number of reads in millions, and then you can see that cDNA with nanopore and CapTrack with nanopore, for this we were able to obtain like 20 million reads approach per sample, while the other methodologies we were only able to get like 2-3 millions in each case. So very big difference in the number of reads. And the other big difference was the quality, so which is indicated here. In the same way as the long reads, the quality was better for R2C2 with nanopore and PacBio. The PacBio in general gave higher quality than the other nanopore technology. So you can see that we have differences in read length and number of reads, but also in the quality of the reads. Okay, so this is the data that we put to the community. We had in total 6 library preparation and sequencing combinations, 3 samples. We decided to have 2 analysis options, only using the long reads in combination with short reads. That gave us 36 data analysis possibilities. And what we did was to put this in the public domain and ask developers of tools to use the data to predict transcript models and submit the results of these predictions. So in each case, we wanted that the analysis was done by the tools developers so that they could give us, the best results that could be achieved with the tool. So in total, we had 11 different tools who participated in this challenge. And I make here a difference by how these tools work differently. There are a number of tools, Bamboo, Flames, Flare, and IsoQuant, that are heavily based on the reference annotation for the transcript model predictions. Some other tools use this information, but not as much. And we have here IsoIB, IsoTools, Mandalorian, Spectra, and Talon. And Lyric does not use the reference annotation at all. So 11 tools participated, not in all the 36 data analysis possibilities. Each tool selected a number of them. And in total, we have 47 pipelines, which is the combination of the library prep, the sequencing platform, and the analysis tool. And this is what we evaluated here. Just to give you some feeling of how the transcript models resulted, just by looking at the technologies. Here you have for the transcript models across the different submissions, differences that we have in length, number of exons, and counts per million. So here you can see how it looks like for the predictions that were using only PacBio data in in yellow, only nanopore in orange, and both. And you can see that the length and the number of exons of PacBio was in general larger than only with nanopore, and the ones that were detected by both were kind of the middle, as you can imagine. But also we saw that in general, the PacBio transcripts were detected with fewer counts per million than those that were detected with nanopore. And if you then look at the library preparation methods, you see also differences. The R2C2 and the cDNA is where we had the longest transcripts with more exons and detected with fewer reads. So if you compare this with the information that I gave you before on the quality of the data, so it seems that the length so the longest transcripts were obtained by those technologies in which you have the longest reads and also the better accuracy of the reads, and not necessarily the larger number of reads. So that was one of the conclusions of this effort. So to evaluate then the quality of the data, we use the SQANTI tools that I mentioned is what we develop in my lab. SQANTI is a pipeline for quality control. We also do some filtering and annotation. We use just the quality control in this project, and this is what I will explain here. And one of the things that the SQANTI does is to create these, what we call SQANTI categories that is a way to compare the long-read transcript models to the reference annotation. So we have a number of classes that are kind of very much used now by the community. Full splice match are these transcript models in which all the splice junctions match perfectly the reference transcript, as you can see here. Then we have incomplete splice matches when we have some missing junctions either at the beginning or the end of the transcript. This could be truncated transcripts or alternative transcription start sites or termination sites. We have novel in catalog. These are transcript models that are a novel combination of existing donor and acceptor sites. So basically, you have a different combination of exons, or for example, like here, you have this junction that is not in the reference, but it's just representing exon skipping, and it's a junction that is using these donor and acceptor sites that are already annotated. And then we have the novel in catalog, in which we have a completely new splice junctions, as you can see here, is not in the reference. And then some other things like genic intron, what is in an intron, genic genomic, which is both intron and exons. We have some other things like fusions and antisense, but usually these are not really very abundant in this type of analysis. The other thing that SQANTI does is to evaluate how the transcript models are supported by external data. And in this project, we have cage data that is indicative of the transcription start site, quant-seq peaks that are indicative of the termination sites, and we also have the short reads by Illumina that give us some information about the junctions, so we can see the coverage of the junctions. Okay, so with this, we evaluated the submissions, all these 47 pipelines. And here I'm represented first, impression, which is the relationship between the different SQANTI categories. And I will walk you through these graphs because they are a little bit complex. So here you have the different tools with the names, and then in colors, we have the different library preparation methods. So if we look at the full splice matches in comparison to the incomplete splice matches here, you can see that there are a number of tools. These are those that are heavily based in the reference that have a lot of full splice matches, very few incomplete, and also very few of the novel categories. So these are the tools that are heavily based on the reference. Those tools that are kind of using the reference as a hint, but not really that strong guidance. They are kind of an intermediate position, so they have a less number of full splice matches, which are the reference transcripts, allow more incompletes, but also more novel transcripts, especially if the novel in catalog, these novel combination of existing splice sites. The lyric tool that is agnostic is the one that is detecting the largest number of novel node in catalog. And one thing that was interesting here to see is that actually our results were more dependent on the tool, right? Here. So on the tool that on the actual data. So the data from the same tool with different types of the prediction for each tool with different type of library preparation methods clustered together in this representation. So in general, tools that provide a high number of full splice matches are strongly based on predictions that use the genome annotation. But we could also see, because we have these experimental data like CAGE and QuantSeq, look at the experimental support of these sites. And this is contrasted with the support that we have by the reference at the 5' end of the 3' end. And again, you can see that these predictions that were produced by the methods that are heavily based on the reference, they tend to have a very good match with the reference, logically, but like a medium support by the orthogonal data here in the case of the CAGE, maybe they go to 60, 70%, and especially the support by QuantSeq data and the transcription termination site was lower than the rest that are better supported by the experimental data than the actual reference. And again, in Lyric was at this site in which the support by the orthogonal data was high, but not that much by, but not as much as by the reference annotation. So basically what this is telling us that the tools that use the reference annotations are actually a bias of more supported by the reference than the actual experimental data. So this is suggesting that in these cases, we are having an overcorrection of the transcript models for this type of tools. So this gives you a little bit of an impression of how the tools work, but some other things that we looked was at the overlap in transcript models. And here you have the number of transcript models. In this case, we call this unique junction chains. The numbers that is detected by an increasing number of pipelines. And this is in logarithmic scale. And here you have the SQANTI categories in these numbers. So what you see is that the majority class is those that is detected by just one pipeline. So these are the 47 pipelines, right? And this is in the log scale. So there are really many transcripts that were only detected once. While the number of transcripts that were detected in all cases were very few, or something like 10 or 20. And also you see that in these cases that transcripts are detected by few pipelines, we tend to have most of the transcripts be novel. So the full splice match, which is the known transcripts, is increasing as the more pipelines are detecting this transcript. So what we are seeing that pipelines tend to agree on the known transcript, but not on the novelty. So they don't usually agree in the novel transcripts. And if we try to see where the consistently detected models are and how is the structural categories, this is what we find. We find that actually if we look at those transcripts that were detected at least by 3 methods and 3 different samples, most of them will be the full splice matches. But there is a number of them. This is the novel in catalog category that are actually consistently detected. And we think that this will be like the core of the transcript of the new transcripts that can be detected by the different methods. And the tools that are giving these results mostly Bamboo, IsoQuant, Flare, and maybe also Mandalorian. Okay, so this was a kind of indication of how these look like in terms of SQANTI categories and agreement, but actually we don't really have a perfect ground truth here. So for that, we use these spikings, the SIRFs. So these are RNAs that are put in the reference, in the sample, and we know exactly how they are so that we can calculate sensitivity and prediction. So using the series, and in this case, the information of the annotation of the series is known to the tools. What we can see is that in general, most of the tools were very good at sensitivity, especially with PacBio tools that are the orange and the red ones. Lyric and Talon were struggling a little bit on more on the sensitivity. And also the same happens with prediction. And only in some cases you have lower predictions for some nanopore library preparations. And again, Lyric and Talon had poorer results, right? But in general, the others had good sensitivity and precision. These were generally short series. We also have very long spikes, and this is the result. So it's not as beautiful then. So there are some tools like Flames, for example, that is not able at all to detect the longer transcripts, and some other tools have more difficulty to find them. Tools like, for example, Bamboo and IsoQuant is actually giving a very good result in these cases, right? And but you see there is much more variability. And one thing that is happening here, for example, if you compare Bamboo with FLAIR, is this, what I'm saying, this overcorrection of this tool in contrast to other tools that are really looking more at the data. Because if you look at these long series, you know, have 4 kb, 6 kb, what you see is that this is indicating how well they are covered by the long reads. You are seeing that in most cases after 4 kb, it will be hard that the transcripts are actually well covered by the long reads. And the only one that is giving the best coverage, as we already know, is PacBio, but the others really have important gaps in the coverage of the long reads. So currently, you know, this will be the technology that allows you to capture these longer transcripts. Okay, so because here we knew which were the transcript models, we actually cannot really say much about how is the accuracy in detecting novel transcripts, which is one of the, you know, of the claims and one of the promises of the technology. So to be able to evaluate this, we use computationally simulated data, both for PacBio and for Nanopore. And using this simulated data, we also assess the pipelines. And here you have, this is now a different type of representation. This is a radar plot in which you have the different pipelines. We look at sensitivity for highly expressed or more expressed transcripts, and precision. These are the known transcripts. And then we have sensitivity and prediction for the novel transcripts. And this indicates the redundancy. So how many times the same transcript models is predicted for simulated transcript. I will comment the results with PacBio that are in red because the nanopore simulation was giving funny results or not as good. But what you can see here, for example, if you look to Bamboo, is that this set of tools that are heavily used, the reference, they are really good in giving you a good sensitivity and precision, as we knew for the known transcript, but then they are really having very medium precision and sensitivity for the novel ones, right? This happens here, also with IsoQuant, especially for the sensitivity. And in the case of FLAIR, what you see is that here you have two different library preps. The darker one is what's the library preparation, the analysis in which you are using also Illumina data, and you can see a big improvement in detection of the novel from not using the Illumina data and using the Illumina data. Some other tools with Talon was not very good for the novels. And then we have Mandalorian that was also of the best performing tools regarding the novel transcripts. Another tool, for example, Spectra, gives you a high sensitivity, so it's able to detect a lot, but actually the precision is bad, indicating that there are a lot of high false positives, and this happens with Spectra, ISO tools, and ISO-IB. So they detect many, but also many false positives. So one thing that, so here then you can see already some differences in the ability to detect these novel transcripts. So here, this simulated data simulate a sequencing error, but not as well the library preparation. So for doing that, we did this manual annotation with gene code. So basically what we did here was to select 50 loci, and we gave it to gene code curators who did a manual annotation of the different experimental data sets, meaning library preparation and sequencing platform. And what GeneCode found is that there were really a lot of novel transcripts that were also found in only one experimental data set. And that was really surprising. So you can see that the majority of the transcript models were actually novel transcripts. But if you look at them, at those transcript models that were called by gene code curators in the 6 combinations of library preparation and sequencing platform, they are all novel transcripts, right? So the novel transcripts are detected in just 1 or 2 data sets. So indicating again that we have really here a lot of sample-specific transcript models that actually validated by the expert curators. So we took these data sets and asked by the different tools how well they were able to predict. And again, we differentiated the novel, the known, and the novel transcripts. And what you can see here is probably what you expected. So in general, for known transcripts, detection was kind of more or less okay for tools like the ones that we knew, Bamboo, IsoQuant, was good. Mandalorian was also okay. Flare was good at precision, but not as well as sensitivity. These were, in general, transcript models that were more lowly expressed. But, you know, there was a pretty good performance in this case. However, when we look at the novel transcript models, the tools in general really failed. The sensitivity was generally very bad. And the precision was only good in those cases where just a few transcript models were detected that were correctly detected. So what we saw here is that there is still a big challenge by these tools to accurately identify novel transcript models. And so there is much work that has to be done here. And then finally, what we did was to do some experimental validation of this. So we took a number, 100 transcript models, and then we validated them by RT-PCR. First, what we did was to look at those that were detected by gene code, and then we could validate by real-time PCR most of them, both the known and the novels. Some of them were rejected, but and we validated them, but these were like mapping errors. So the transcript was there, but the transcript model annotation was wrong. But then with the ones that were detected by the different categories, this is what we found. So those transcript models that were consistently detected by several, but many pipelines, many samples, either they were full splice matches, novel in catalog or novel not in catalog. So the consistently detected were validated in almost 100% of the cases. So we have a very high validation rate of those transcript models that were consistently detected. For those that were rare, what was just associated to 1 or 2 pipelines, what we find was also surprising because the novel in catalog, so they are novel combinations, they were also normally detected. But, and those that are the novel not in catalog were kind of detected only half of them. So if there is a lower validation rate of this part, but actually there were quite a number of them that were also validated. And this was surprising because it indicates there is a lot of transcriptome diversity that can be actually validated, but it's very hard to capture by the different approaches. We also look at, in how many samples we need to find transcript models to be able to validate it. And yeah, if you have it in more than 2 or 3, most of them will be validated, but also just with one, like we have around 70% validation rates, indicating that we find a lot of novel transcripts that are sample specific, but they are validated. So the conclusion of this part is that the long-read sequencing can help you to identify transcripts with, I would say, great precision in general, but actually the results depend more on the algorithm than the actual data. So for the calling of the known transcript models, at least, globally, PacBio with cDNA and Nanopore with R2C2 are giving the best results. And here you have a number of tools that were performing well with the known transcripts. Precision was better than sensitivity in general, but consensus in the discovery of the novel transcripts is still very challenging. However, we see that these can be validated. So there is, some work to do here to accurately detect novel transcripts. So, so this is about quality control and technology. So I will try to go quickly through these 2 other aspects that I think that are also important, that are related to the functional annotation and the analysis of the data. So we created this ISOAnnote tool to try to get a functional annotation of at the transcript level and with isoform resolution. So basically, because we are detecting all these new transcripts, we really cannot, we need to do something to say something, we need to do something to assign function of at least potential function to these sequences. And the ISOAnnote tool, what it's doing is just to put a large number of different tools and methods for the identifying functional sites, functional motifs, domains, both on the RNA sequence and on the protein sequence of these transcripts. So we are using a combination of predictors, for example, to predict upstream open reading frames of polymerization sites, regulatory elements at UTRs or repeats. We are also using information from ClipDB and Milwork to annotate microRNA binding sites and RNA binding protein binding sites that we transfer from these databases to the transcript model. So this is at the RNA level, and then at the protein level, we are also doing the typical thing of mapping the domains, localization signals, disorder regions, transmembrane regions with predictive algorithms, and then we use databases for other type of things like the post-translational modifications, look at localization signal that important active sites and other type of sites, right? So we have this combination, and in general, we are achieving a very high level of functional annotation. This is all put together, in a SnakeMake pipeline that what is trying is to connect the different tools and give an output that finally we can use for doing functional profiling of the transcriptomics data. I will not talk too much more about the ISOAnnote because I want to go into how we are using the information. So we developed this tool, which is called tappAS, which is user-friendly tool with a very nice graphical interface to do differential expression analysis and functional profiling of long-read resolved transcriptomes. And here you have some of the screenshots of the application. And yeah, I can give you some examples of how we use this. My example will be given on these data sets in which we were profiling the differentiation of neural progenitor cells in mouse towards 2 different lineages differentiated into oligodendrocytes of motor neurons. And this is something that we sequenced with PacBio and we also have Illumina. Well, yeah, we have data for both lineages. Most of the things that I will be showing corresponds to comparing these neural progenitors with the differentiated oligodendrocytes. So one of the things that we can see from the functional annotation and with tappAS is the intensity of the functional annotation strategy for the different type of databases that we use. So what you have here is a representation of the genes that have multiple isoforms. So that will be the 100% of genes with multiple isoforms. And then what is indicated here is of all the isoforms in the genes, which is the percentage of genes for which the isoforms have different annotations in that particular database. For example, just to give, for example, if we talk about post-translational modifications, right? So this will be this bar. So we have that about 60% of our genes with multiple isoforms had annotations in this database, and more than half of these genes had diversity of annotations across their isoforms, meaning that if I have a change in the expression of the isoform, I will also have a potential functional change because the functions annotated are different. So what you can see is that, and then we have the databases that are analyzed at the transcript level that relates to protein properties in blue. So what you can see here, for example, the length of the UTR. So yeah, the length is something that we could always establish, but you can see that in the majority of the cases of the transcripts that have multiple isoforms, there is variation in the UTR length, right? And also in the coding sequence, or also in the presence of upstream open reading frames. And at the level of the protein, we also have quite some diversity. For example, these are PFAM domains. You see that the majority of the genes in which we could annotate PFAM domains have different annotation across their isoforms. So meaning that studying changes in expression of isoforms may be having a functional impact. So this is the type of analysis that it's done in tappAS. We have the traditional analysis like, differential splicing, differential expression, what I just saw about this variability analysis of the annotations. The input data in tappAS is the gene models that you obtain, for example, with the long-read sequencing, with the annotation of the isoforms, all this functional information identified by isoanode, and then quantification metrics of the expression of each isoform in one condition. Initially, we were quantifying with Illumina reads. Now you can quantify directly with the long reads. And then we do things like I mentioned, differential isoform usage. We do functional enrichment, but we also have a number of analyses that are specifically developed by this tool that is trying to identify the different inclusion of functional features. For example, a differential feature inclusion will indicate whether a functional element is differentially included when you compare two conditions because of how the expression of the isoforms is controlled. We can also look at differences in polyadenylation sites. We can also see whether there are some features that are co-included or co-excluded. We can also evaluate differences in the change of the UTR lengths between conditions and so forth. So here we are focusing on the functional elements, and here we are focusing in the isoform itself. So things that we can do, as I mentioned, in this traditional analysis is that to use differential expression and differential isoform usage and the switching. So here you have two conditions, right? And this is a gene with 3 isoforms, and this is a representation of this orange isoform, goes from being lowly expressed in the cases to being high expressed in the controls, for example. This is a differential isoform usage. And then we also measure the magnitude of this change. This is what we call the total isoform usage change. So how much the relative percentage of the expression of the isoform is changing from one condition to the other. So, and then one things that we can see is to compare the changes in the expression of the gene expression. So the total changes in expression between two conditions, it will be the log2 fold change, the traditional one, versus this total isoform usage change. And then what you can see is that there are genes that are, for example, only regulated by changes in expression. These are the ones that are here. And also there are other genes that are both regulated by changes in gene expression and changes in splicing. This will be the ones with the blue dots, right? And there are a number of them, for example, these ones here, the ones that are here in the middle and on the top part, that actually are only regulated by changes in the expression of the isoforms. So these are genes that you will not capture with the traditional differential gene expression analysis, and only when you look at the changes in isoform usage, you can see that they are regulated. And then what we can do is to take these genes that are regulated by differential isoform usage in blue or differential gene expression in orange and look for functional enrichment and see how each functional category is actually regulated by both types of processes. And you can see that most changes in this oligodendrocyte differentiation is regulated both at the level of gene expression and at the level of splicing. And most of the times is the gene expression which is driven the changes, but you have some cases, for example, here in brain development of myelin cells, that the chromatin, the nuclear chromatin, that are mostly regulated at the level of differential isoform usage rather than at the level of gene expression. So again, you can identify here how each cellular process of biological process is regulated either transcriptional or post-transcriptionally. And here I'm giving you some kind of specific examples of things that you can see. For example, here you can see the one particular case, which is the alternative splicing of the MUL1 protein. And here we have a case in which we have a change in expression that is being associated to the differential inclusion of a transmembrane region. And here the difference is between oligodendrocyte and motor neurons. And what we see is that this transmembrane region is actually included, so in red, in the motor neurons, right? When you have the differentiation in motor neurons versus the oligodendrocytes in which you don't have the inclusion, right? So you can see here. So there is a differential splicing leading to the differential inclusion of transmembrane region for this gene. So what is this gene doing? This MUL1 gene is involved in the equilibrium in the mitochondria, is regulating the equilibrium between fusion and fission, because it's regulating some other proteins. It's called OPA and OMA. And what this protein is doing is regulated the balance between fission and fusion, and this is resulting in a control of the energy balance. So finally, what we think that is happening here is that because of alternative splicings, we are controlling, so how is the shape and the abundance of mitochondria. So in oligodendrocytes, you will have the fused mitochondria, while in motor neurons, you will have separated mitochondria, right? So that you will have the split mitochondria, and that will provide the increased energy that is needed for this particular cell type. And with that, I want to come to final conclusions. As I mentioned before, defining isoform models from long reads is possible but challenging. We have developed the tools to create these transcript models and annotate them functionally, and also investigate the functional consequences. We found this large number of samples that are systematically used in individual samples. And that should make us reflect on how to define and annotate the transcriptomes, right? So we maybe want to ask questions whether each single molecule deserves to be called an isoform, of how we develop tools to annotate this pool of RNAs. And I want just to finish indicating that I'm participating in this special issue of genome research on long-read sequencing methods. And I just heard that they extended the submission deadline. So if any of you are working with these long-read technologies, both for transcript annotation of analysis, but also for genome assembly of any kind of other application, so you are welcome to submit your work to this special issue. And with that, I want to finish by saying thanks to the people who contribute to all this work. My lab, most of the work that I am showing here, it has been developed by people that are not there anymore. But especially, as I mentioned before, Fran Pardo was very much involved. Some other people, Lorena de la Fuente and Pedro Salguero, were working on the tappAS tool. And now most of the people working on long-read methods, and especially in the analysis of this strange of rare fraction of the transcriptomes are Caro Monzon and Alejandro Paniagua. And with that, I want to thank you for your attention and take your questions.
I have a mic to pass around, so if you want to ask a question, please line up here. Use the closest possible way to just ask your question.
Anyone in the room?
We have also from the Zoom.
Oh, we have a question.
Can you?
Okay. I have a voice for you. So on the slide 25, you talked about experimental validation of the novel transcripts that were detected across multiple platforms. Can you explain how that was done? Is that looking at spiked in or synthetic data, or was it a targeted sequencing done to look closer at the novel transcripts?
Yeah, that was a targeted sequencing. What we did was to design primers that will amplify specifically the transcript models that we detected. And then we went to the samples and then used PCR to amplify those transcripts, and then we sequenced them again, both with PacBio and Nanopore. And then we check whether we have found back the transcripts that were predicted initially. So targeted resequencing.
Oh, I have a follow-up question on that. So in the case of direct RNA sequencing using Oxford Nanopore technologies, so which combination did you find the most effective approach?
With Nanopore, the most effective approach was the combination with R2C2 library preparation method.
Mm-hmm.
This is developed by the Wolmer's lab. And basically what they do there is that they create a kind of circular molecule of the transcript that is then amplified by PCR.
Mm-hmm.
And then, well, keeping being circular, right? So, and then like the amplified molecule, the amplified transcripts is sequenced with Nanopore. So what you achieve in this way is that the transcript is actually sequenced several times through the pore,
Mm-hmm.
and that you have the information that is the same transcript. It's trying to replicate what PacBio does in a way. So then you do a consensus call of the transcripts, and that's increased the accuracy. And what you achieve with that is, much better results. So what we found is that for detecting good transcript models, is more important the quality than the quantity.
Cool.
Thank you for the great talk. So one of the things we actually worked when we were working on, detecting novel isoforms was that we had a matched sample, long reads as well as Illumina, but we had a high coverage Illumina short reads, like 80 million to 100 million reads. And we had used a single flow cell for long reads, nanopore. So we encountered isoforms that we actually see a junction exist, right? And I'm not talking about retention, just, if you consider the annotation, the junction exists. So we were just going to validate some of these, but when we were looking at the short reads, we didn't see any evidence that actually this connection, because it's a junction, you should be able to see a read or two. We didn't see this happening. So I'm wondering when you were looking at the consortium data, like, did you have these scenarios? Like you had also, I think, short reads as well for some of the samples, if I'm not mistaken. And if so, is there any explanation biologically what could be happening over there? Because, I mean, they are the same, we use cell lines, actually, we use cancer cell lines. So we sequenced it, and we using even the same plates. So we had just split it into two, just prepped it and sequenced it. So any insight if you have encountered such a phenomena?
Yeah. So yeah, absolutely. So we found that, and that was that was yeah, that was usually a quality control element, right? So and we usually will see that with nanopore we have less support by Illumina reads than with PacBio, for example. And things that we see, for example, is that the full splice matches, so the transcripts that are known in the reference, tend to be very well supported by Illumina reads, while the novel not in catalog tend not to be as well supported. So this is already indicating that, that you have artifact of the technology, and this is what you are sequencing. One thing that I have to say, though, is that we are recently using the new maSigPro of Kinnex data from PacBio that is now producing, it's given us like 40 million reads, PacBio reads, right? And then when we go there with our Illumina reads and try to find the support with Illumina of the short reads, with the Illumina short reads of the junctions that we find with the maSigPro, we don't have. We have like a 50 or 60% validation rate, right? But then what we did was to use the recount database. So the recount database is a collection of all the Illumina reads that have been published through the years, right? That is the largest collections of, transcriptomics data that you can find. And then when we took this information, then we kind of validate most of our junctions. So what this is telling us is that we are reaching a moment, and I don't know how many nanopore reads you had, we are reaching a moment in which actually, if you count the sequencing output that you have with the long reads, it's bigger, it's larger than what you get with the short reads. Because even if you have many short reads, they are short, right?
Yes.
So actually, there will be a moment in which, I don't know, again, I don't know how many reads you got with your nanopore.
We got about 12 to 14 million reads. They were cDNA, not direct RNA. So
yeah. So if you make the maths, you will see that the amount of sequencing output probably, I mean, it depends, right, on the length, but the amount of sequencing outputs maybe that you have with nanopore is actually larger. So it's not surprising that there will be some junctions that you don't find with Illumina.
So basically, it's saying that if accumulative lengths of the short reads equal to accumulative lengths of the long reads, then there's a chance that you will see it because you're using the public data sets and you are seeing because you're increasing the total, I mean, I'm not going to coverage, actually throughput.
Exactly, yeah.
Okay, that's great, actually. Thank you. Any other question at UBC side? Okay. Oh, there's one more in the chat. Okay. Bada, can you ask your question? I think you can unmute yourself and ask your question.
Oh, yeah. Thanks so much for the great work and really comprehensive talk. I mean, disclaimer, I'm Faraz's PhD student, and we've been working on Freddy, which is annotation-independent tool for long-read isoform detection. We started a bit late, so we definitely missed the long-read.
So one thing that we have, maybe I invite you to try it, and if it's working, you have to tell me because it has been a little bit problematic. But we have put all the data and the evaluation, scripts. Yeah, the evaluation pipelines in an open eBench platform so that you can take the data, use Freddy for annotation-dependent annotation, and then put it through the open eBench, and then you will be evaluated in the same way as, the longer GASP, and then you will see how you compare with the rest of the tools, right? So the annotation independence part of the challenge was really, really bad,
Yeah.
So there is a huge need of tools that are working there. So please do that. And we have been having some kind of trouble to have the open events things working smoothly. The challenge 3, which is doing the reference-free evaluation, should be working fine. So if you want to try, let me know if it works, because we need beta testers.
Yeah, definitely. Then I think you'll be getting an email from me soon. Sure. I guess like question regarding the evaluation methodology overall. I mean, this is something that I also like struggled with when like building like such a tool, is the question of simulation, and how critical do you think that building in a simulation to reflect the library preparation issues, and maybe specifically truncation, PCR artifacts, maybe chimeras even, in like being able to really evaluate how these tools work?
Very. I mean, the simulation, so I believe that the problem of the long reads is not the sequencing. I mean, nanopore is worse than PacBio, but the problem is the library prep. So the issues that we find is not the sequencing, it's the library prep. And then you have one thing is the truncation, as you say, right? So one thing is that some of the simulation tools try to simulate the degradation, right, of RNA. But I don't think this is properly captured. But other things, like for example, intraprimate, this is something that we see, right? Other type of, I mean, PCR artifacts, maybe that is captured by the sequencing, by the error rate, and this could be more or less modeled. But I think that we need better tools to simulate the complexity of the library preparation methods. Because in general, with the simulation data, we had much. So one of the things that we did in Undergas was to use all these different benchmarking methods. So we use, the real data, the simulated, the SIRFs, the gene code, And the conclusions were not always consistent, right? And then you have to look a little bit above and try to conclude what everything is telling you like a whole, right?
Yeah.
But yeah, I think that we need better methods to simulate data and to go into the library prep. Sequencing accuracy, PacBio is fine. Really, believe me, it's fine. It's so okay.
I mean, I imagine also Oxford Nanopore, if your goal is alternative splicing, the error rates locally is not the problem.
Yeah, I mean, if you want to define transcript models with nanopore, forget it.
Yeah.
No, I don't believe nanopore for that. I'm so sorry. I mean, I love nanopore. They're my friends, but I have to be honest. I think it's great for other things. I mean, I think if you stick to the reference, if you just want to identify things in the reference and then you are just looking for the contiguity and then you correct and you use Bamboo, for example, then it would be fine. But for novel, and there is a lot there, and there are a lot of novel transcripts out there. It's true that they are low expressed. So it's not if you I don't know if I have 4 minutes because I don't know if I have this. Let me see somewhere. No, I don't have it here. But so all these novel transcripts are very low expressed. So the huge majority of the transcriptional output are, I mean, at least in human and mouse, are perfectly defined in transcript models and are highly expressed. But then you have a tiny fraction of things that are, I call this RNA biological noise. So they're true, but they are very low expressed. They are sample specific, and some tools keep them and then create different transcript models, and everybody is doing a different thing.
Yeah, thank you so much. I don't want to take more time from the question.
Yeah.
And hopefully, yeah, we'll talk after.
Sure.
One final call for any question from either sides. If not, thank you again, Dr. Conesa, for
thank you for your invitation and good luck with your transcriptomics analysis.
Thank you very much. Let's
Bye-bye.
one more time.
Take care. Bye.
Bye.